Good morning to all. Uh, it's my pleasure to introduce my colleague, Professor Suvarn Kulkarni, who is excellent synthetic chemist, especially in carbohydrate chemist, and is also excellent teacher, one of the excellent teacher in the department. He has obtained his MSc and PhD both from Pune University, graduated in 2001, and then he had a long postdoc in Economica Sinica Taipei and University of California, Davis. And before joining IIT Bombay, he joined IACS Calcutta for a short period and joined this department in 2009. And from 2016 onwards, he's a professor and is also an institute chair professor. And he, of course, received several awards that includes Professor S.P. Sukatme Award for Excellence in Teaching and CRSA Bronze Medal, and is also a Fellow of Royal Society of Chemistry, London, and is also an editorial member of the Journal Carbohydrate Research and so on. And with this brief introduction, I request Professor Kulkarni to deliver his talk.
Thank you, Professor Ravikant, for a very kind introduction. And also would like to thank IRCC for giving me this opportunity to share our work today. Let me share my screen. Okay, are you able to see my slides?
Yes, if you can make it full screen, that would be nice.
Yes. Thank you. So the topic, title of my talk today is the same topic of our paper. And we are really grateful to IRCC for giving us Research Dissemination Award. This is a huge honor and I really feel humbled. And today, so basically I will describe what we had given in this review and what were the future perspective in 2014 and where is the field. So this is the review, recent advances in synthesis of bacterial sugars, building blocks and their applications. And it has received very good citations and applaud from the scientific community. And in last 7 years, so far, this has remained the only review in this field. And this is what we had said in the review. So very simplistic manner, we had said that the bacteria have very unique sugars on their surfaces, which are not present in human hosts. And these sugars, they play important roles in variety of biological events, and one can use them for therapeutics and vaccines. And how do we do chemical synthesis? So this was the main focus of this review, is what are the synthetic approaches available at that point of time, and how we can extrapolate that, and how we can develop one-pot synthesis to make longer oligosaccharides. So this was the content of the review. So let me begin giving you a brief introduction of this field. So infectious diseases are on the rise, and they have continued to threaten human life constantly. And the most relevant example is the recent COVID-19 pandemic, which all of us we have experienced. And what have made things more complicated is this antibiotic resistance, in which he talked about it yesterday. Also, Anirban mentioned about some pathogens. So the question is, can we develop novel antibiotics? That's one approach. Or how do we overcome this antibiotic resistance? So can we develop antibiotics, therapeutics, vaccines? And our area is carbohydrates. So we asked this question, that how can we develop this therapeutics and vaccines from carbohydrates? And how chemical synthesis can help us to reach this goal. So why carbohydrates? Because carbohydrates, as you know, they are ubiquitously distributed on this cell surface, where they participate in a variety of vital life processes such as viral and bacterial infection, cell-cell communication, and immune response, to name a few. And they're diverse and complex, and they are present in microheterogeneous form in nature. So if you try to isolate them from natural sources, you won't be able to do it in pure form and acceptable amount. And that's where synthetic chemists like us come into picture. And here is one more cartoon showing you the exact position of carbohydrates, where we roughly classify them into glycoproteins or glycolipids. So these carbohydrates are attached to proteins on the cell surface, whereas glycolipids are directly attached to the cell surface through some linkage. And as you can appreciate that, just by virtue of their position on the cell surface, they are the ones who talk with other cells, right? And for many years, until year 2000, it was believed that glycosylation, which is a post-translation modification of protein, is only present in us, in higher cells like eukaryotes, and is virtually absent in bacteria. But in last 15 years, things have changed, and now it is well accepted that bacteria also have glycosylation pathways and they have glycans. And it was clear from this TM image, as you can see, this is a bacterial cell, and these appendages, you can see up to 50 to 90 nanometer in width, is totally covered by this glycans, the glycoproteins. So bacteria have glycans, but there is one difference. While human, we need maybe 10 sugars to complete our glycan. Bacteria need hundreds of sugars, and there's a huge diversity. And there's a select group of bacteria which have such kind of deoxyamino sugars. So if you compare with glucanac and galanac, the normal sugars, these bacterial sugars lack this oxygen here, and they also modify this particular position. So we focused in this review on this select group of compounds, and we described the methods available for synthesizing these compounds and the applications. And since these are present in select group of bacteria, we thought that they will be very helpful for detection of pathogens and for selective targeting of pathogen. And to just give you a few examples, for example, this one from Neisseria meningitidis has this sugar has diacetoaminotrideoxyhexose, or this longer glycan in C. jesuni, this has a bacillozamine. This one from Pseudomonas aeruginosa has fucosamine, and some deuterionic polysaccharides from certain bacteria have this sugar called as AAT. More importantly, the presence or absence of these sugars has been shown to be linked to pathogenesis. And because bacteria have these sugars and we don't have, they are excellent candidates for target-specific drug delivery and, more importantly, for vaccine development. And what is required is, because we cannot isolate them in pure form from nature, and we need a lot of amount for drug discovery, what is required is you need chemical synthesis to make these compounds. So this was a background of this review, and then we described synthesis of these compounds. And you will be surprised that in 2013, when we started working on this project, we did a literature search. And this is what my first student, Madhu Emadi, found from SciFunder, and I was shocked. This was an exhaustive list of references. So we were looking at just 10-15 references in literature where people have made some efforts to make these glycans. And this is an exhaustive list of references. And what was important is that all these methods described, these are all big labs, and they had synthesized this compound using longer routes, 20-step synthesis, minuscule amount diastereomeric mixtures. Then Seeberger's lab started publishing de novo approach, where you start with a smaller fragment, let's say 3-carbon chain, and then you decorate into sugar. This was also giving a diastereomeric mixture. So we realized that there's a gap in literature, and we need to develop, we have a scope to develop a method. But if you're starting from carbohydrate, we should do like enzymes do, very specific reactions. And this was our idea at that point of time. So we thought that we can start with simple sugar mannose. We will do selective deoxygenation because this is more reactive. Then on the triol, we will use metal coordination and take help of the same side disposition of these 2 hydroxyl groups to mask this hydroxyl. And then on this diol, we can convert them into good leaving group and then do selective displacement of one of the leaving groups to get access to this. And then in the same pot, we can add another nucleophile. We can vary the nucleophile, and we can get access to all these sugars in one pot manner, short method, fast method. That was the idea. And indeed, I don't want to give all the details due to lack of time and due to the nature of the audience. But suffice to say that starting from this sugar, Madhu was able to establish this protocol to get access to all these sugars. And this worked really well. So from this diol to all the rare sugar building blocks, he could go on a gram scale in excellent yield and just have to do one chromatographic purification. So with the success of this, what we did is we could then, our lab and other labs took help of this method and synthesized a variety of glycoconjugates which are useful for vaccine development, and then I'll just show you some of the glimpses of that. So this trisaccharide from Neisseria meningitidis, we synthesized this, and then this was pseudo trisaccharide from Bacillus cereus. Then this was from Providentia alcalifaciens, and they have all these strange appendages which are difficult to introduce, and this has all these rare sugars AT in this, and also more recently this deuterionic polysaccharide unit of Bacteroides fragilis. Also, Madhu was able to synthesize this hexasaccharide, which has all even more complex sugars. And then more recently, our lab has synthesized these two from Pseudomonas and pneumonia, pneumococcal serotypes, and this one is from Streptococcus pneumoniae. These are the recent works, and we are still in the last phase of completing the synthesis of this Vibrio choleric glycan, which has even more complex sugars like viosamine attached with the amide, and this one has a bacillosamine. So then we asked, can we really go for application? So around 2017, WHO for the first time published a list of highest and critical priority pathogens. These pathogens were those pathogens which had developed this antibiotic resistance. So you acquire these pathogens during your stay in the hospital, and they are the causative agent of nosocomial infections, and they are multi-drug resistant. So can we develop a vaccine against this Pseudomonas aeruginosa, and who gave a list of pathogens called as escape pathogens. So this one from Pseudomonas aeruginosa, and we need a linker for conjugation to protein. So this is Staph and which are very difficult to synthesize. And to cut the long story short, Archana and Diksha from our lab, they were able to use our protocol to develop these two building blocks which they could tie together stereoselectively and carry out total synthesis of these two glycans which are ready for protein conjugation, and then immunological studies which we are doing in collaboration with Peter Seeberger at Max Planck. And this one more compound, this has all rare sugars. This we are also evaluating the immunological properties of this. This is from for stomach infection. This from Placimonas shigelloides. So the access to rare sugars enabled assembly of these glycans for vaccine development. And in last 2 minutes, I will just give you what others have done. So this Cody from Netherlands, he used our method to make these AAT building blocks and synthesize really longer glycans from Streptococcus pneumoniae. And then he showed that 9 mer or 12 mer are more active. And these kind of studies were not possible before due to the lack of access to these kind of compounds. Recently, Chauffet from Michigan State also used our method to construct some longer glycans from pertussis, and they are developing glycoconjugate vaccine for pertussis. Okay. And then in last couple of minutes, I will show you how you can use these compounds for development of narrow spectrum antibiotics. See, when we take antibiotic, we end up killing good bacteria. So our question was that these sugars are specific to these bacteria. Can we use these sugars to reach out to specific bacteria and then kill only those bad ones, leaving the good ones in there? So can we develop a narrow spectrum antibiotic? So this was a paper, if you are interested, you can look up. This was a study done in collaboration with Daniel Dubé in USA. And what we were able to do is using these synthetic sugars having azide as appendages, we were able to selectively label pathogens, only bad pathogens, leaving the good ones intact. So selective detection was possible. And at that point, we thought this is because of the species selective differences in the carbohydrate synthesis pathways. That means bacteria have a different set of glycosyl transferases from us. And that's why we can label them selectively. So the question was, can we use this difference for selective targeting? And very recently, we have achieved, I should say, a proof of concept study, although a lot more has to be done. So the idea was that bacteria need these elaborate glycans for infection and for survival. And can we interfere with the glycan biosynthesis of bacteria so that we can truncate this, and this will result in truncation of it. And this will lead to potential and inhibitors. So with this idea, we developed two types of inhibitors. One is a benzyl glycoside, so the sugar that bacteria has. And then this one is to just, it's like a substrate decoy. So it's to divert glycan assembly. And then other type is the fluoroglycan, whereby bacteria will take this up as a donor, but will not be able to extend the glycan. So in both the cases, we are messing up their glycan assembly, and that will result in the truncation. And indeed, what we found is that that led to growth defects. And so we were able to use this kind of sugars to halt their growth and disrupt bacterial cell wall biosynthesis and halt their growth. So this was highlighted in Chemistry World recently, and we got very good praise for this research. And also thankful to IRCC. This was also published in Research Matters. And then Hindu made a story on bacteria killing sugars. And very recently, even the very popular newspaper, local newspaper, Sakar and Lokmat, they made a cover story. So this appeared on the main front page of this Sakar newspaper. So we are working in this direction. Our group as well as other groups are making the help of our methodology to go ahead. So this is all about this paper, and this was what we said in 2014 in the outlook of the field in the last paragraph. And as you can see, we had hoped that this ready availability of such monosaccharide blocks will lead to breakthroughs in immunoglycobiology, and we expected that it will speed up the development of glycoconjugate vaccine and specific drugs. So with this, what remains to me is to thank the people. So Madhu MRD was my co-author on this paper, and this is a picture taken in 2016. Now Madhu is working in a vaccine company in Germany. And other people also have contributed significantly to this rare sugar project. I'm thankful to my collaborators, Daniel Dubé and Peter Seeberger, and thankful to all the people who are out there using our method. You cannot do anything without funding, and very grateful to IRCC startup grant. Also, when there was a drought season, IRCC helped me with the bridge funding. So I'm very grateful for that. Also grateful to all the other funding agencies, DST, SERB, DBT, CSIR, BRNS, and this was a very nice project, Indo-Israel project from UGC. So I'll leave you with the most recent picture of my lab, and I'm very thankful to all of these students with whom I'm very privileged to get the opportunity to work with them. They are working on these projects. And once again, I would end my talk once again thanking IRCC for this important recognition, and thanks to all of you for your patient listening. Thanks.